In this set of notes, we're going to look at the details of photosynthesis. So again, let's look at the equation. We have 6 CO2, 6 H2O, glucose, and 6 oxygen, and sunlight. The CO2 is used in the light-independent reaction. The water and sunlight are used in the light-dependent reaction. This is our main product, and then this is our byproduct. So as we said before, there are two steps, the light-dependent reaction and the light-independent reaction. We're going to look at each of those individually. So the light-independent reaction occurs in an electron transport chain, also known as an ETC, which is a series of proteins embedded in the membrane that pass electrons from one protein to the next. So it's a series. So if we have sort of like this, with proteins embedded within the membrane, they're going to bounce from one to another. There are two different photosystems. These are the chlorophylls and their proteins in which they're embedded. Chlorophyll, again, being the pigment that absorbs sunlight. They are photosystem 2 and photosystem 1. And we say 2 before 1 because in sort of looking at it linearly, it comes before 1. So here's a picture of the chloroplast, and we see here's photosystem 2. Here's photosystem 1, and these red lines are showing us the bouncing of the electrons all the way through to the end of the membrane. So this is the thylakoid, not the chloroplast. So within the chloroplast, the thylakoid, where the photosystems exist for the light-dependent reaction. So the steps of the light-dependent reaction are as follows. 1, light hits the chlorophyll in photosystem 2. It excites the electrons in the chlorophyll molecules and causes them to jump off. So here we have that light hitting the chlorophyll and then the electron jumping off. The electron jumps to the primary electron acceptor, which is the molecule embedded in the thylakoid membrane next to photosystem 2. So here's photosystem 2, and this is then the first electron acceptor. Go ahead and get the right one there circled for you. Okay, so the electron's going to get excited. It's going to bounce its way, here's the electron, over to this first electron acceptor. The primary electron acceptor passes the electrons to the electron transport chain. So it's going to go all the way through. If you follow these arrows, that's your electron transport chain. Energy from the electrons actually is going to be used in this proton pump right here to pump the H+ ions into the thylakoid, creating this concentration gradient. The light absorbed by the photosystem 1, so this is photosystem 1, which is further down the line, it's being absorbed at the same time as in photosystem 2. So don't think it happens with one first then the other. It's happening at the same time. The chlorophyll electrons jump to another electron transport chain, and the electrons are replaced by electrons coming from photosystem 2. So when the light hits these electrons, those electrons jump out, but the other electrons from photosystem 2 are coming in. So the electrons combine with a proton and NADP+ to form NADPH, and that's this right here. So we have NADP+, and then this is a H+, and what we get at the end is NADPH. This is a carrier molecule. It's going to carry that electron, it's going to carry that hydrogen over to the light-independent reaction, and it also has some energy within the bonds. So the electrons jump out of chlorophyll, but how do they get replaced? Well, that's where water comes in. So water is split by an enzyme near photosystem 2, and here's photosystem 2 in this picture, and we can see water right here is being split. So we have H2O, and it goes to two H2Os go to 4 hydrogen ions, 4 electrons, and then oxygen, and the oxygen is given off as a gas at this point. So that comes out of the system, and there's your H pluses. So ATP is also produced during the light reaction, and it's used to fuel the building of glucose. So it is a product of the light reaction that then goes into the light independent reaction, also known as the Calvin cycle. And it's produced through chemiosmosis. So due to that H plus concentration gradient that we created, chemiosmosis, so movement of something else, a chemical, the electron transport chain has built a high concentration of H plus in the thylakoid, and this causes ATP to be generated through what we call ATP synthase. So the H plus ions, which are just protons, because if you remember, you have one proton, no neutrons, and then you have an electron. This is your hydrogen molecule. But if I take the electron away, which I have done, what I get now is just a proton, which is an H plus ion, but really is just one proton. It's going to move through this ATP synthase, which is a protein, from an area of high concentration inside the thylakoid to an area of low concentration outside of the thylakoid. So once the gradient has been created now, it's going to go through this other protein, and the ATP synthase uses the potential energy that is built by the movement of those protons to make ATP. So the light-independent reaction is the next step, and this is also called the Calvin cycle. And you can see it's in a circle because it is a cycle. So we're going to look at how carbon dioxide comes into play with this 5-carbon sugar that we know as ribisco. And there's another name, we'll get that. And its carbon dioxide is going to be reduced, and it's going to eventually end up down here where we get some glucose being made. But you can see it's going to take some ATP along the way. So let's get started. So the Calvin cycle, which is carbon fixation, it incorporates CO2 into organic compounds. What it does is it takes CO2 and it links it together to make a sugar with other molecules. So 3 CO2 molecules enter the Calvin cycle to make a 3-carbon sugar called G3P. So let's look at how this all happens. The first step, carbon dioxide combines with a 5-carbon molecule called ribulose biphosphate, or ribisco, to make a 6-carbon molecule. However, this 6-carbon molecule is very unstable, so what it does is it splits into 2 3-carbon molecules called 3PGA. So here's your ribisco, or your ribulose biphosphate. Here's your CO2. They're going to come together. They're going to make this unstable 6-carbon that's going to immediately go to this 3-carbon phosphoglycerate, and there's going to be 2 of those. Okay, so for every 3, so in this picture we have 3, we're starting with 3, so we're going to go to 6, okay? So because it breaks in half. The 3PGA is then converted into another 3-carbon molecule called G3P, and this is using the phosphate group from the ATP and then the H plus from the NADPH to make this new molecule, and this process requires energy. So as the phosphates are released from ATP making ADP, we get energy, but we also get the phosphate that we need. And so we get this G3P molecule, phosphoglycerate, is reduced to that, and G3P is going to also combine to, or the phosphoglycerate is going to combine with the H's from the NADPH. So we can see these came from the light-dependent reaction, and they're used right here. So one of the G3P molecules is actually going to leave the cycle and be used to form glucose and other carbohydrates. So we get one G3P gone, and it's going to make glucose when it combines with another. So we need two G3Ps to work to get glucose. However, we don't want to take both of those out because we need to keep a stockpile of those G3Ps in order to make our initial Rubisco. So the other G3P molecules are converted to Rubisco and ribulose biphosphate, and they're going to be used again to keep the cycle going, and this requires energy as well. We have 3 ATP molecules needed to take the G3P and make it into Rubisco. So there are different types of plants that go through these different types of processes with the Calvin cycle. The C3 plants, these are grains. These are plants that only use the Calvin cycle to fix carbon, and they're called C3 plants because they produce 3 carbon sugars. We have C4 plants, or C4 pathway. We find these in tropical plants. These close their stomata during the hottest part of the day, and this is to keep the water in. And they have an enzyme that fixes CO2 into a 4-carbon molecule even when CO2 levels are low. Because when we close the stomata because of the heat, the CO2 is not able to get into the plant as well. So the part of the plant, the stomata, this is just an opening in the leaf. We're going to learn about that. So you don't need to worry about that. That's in another notes area. So the CO2 that's extracted from the 4-carbon molecules later goes into the Calvin cycle. So it's sort of a stockpile of carbon. And then the CAM pathway, this is where we find in succulents, like succulents being aloe plants and cactus and things like that. They open their stomata at night and close it during the day. And so CO2 is fixed into various organic molecules. And during the day, the CO2 is released and enters the Calvin cycle. So it's released from these molecules because the stomata are closed during the day so as not to lose too much water. So these are the different sort of types of ways. You can have carbon dioxide coming in and getting it fixed directly. That's a C3 plant. You can have it being stored in a C4 or a CAM type of pathway.